All right, let's talk about an example of genomic imprinting in humans that's actually coupled with the consequences of having a deletion in a chromosome. So again, we're talking about these 4 rules of Mendelian inheritance and how even though we're assuming that mutations are rare, the genes can pass altered from generation to generation in the sense that they're marked. So there's some kind of methyl group that's being placed onto the nucleotides present within the genes, and that marking pattern can be inherited. Now again, just to remind you of genomic imprinting, these typically follow a non-Mendelian pattern. It occurs when a segment of DNA is marked. This effect is maintained throughout the life of the organism inheriting that marked DNA. Where things change, or where patterns can be reversed or completely removed in terms of the methylation groups or the methyl groups, is in the germline cells. So we refer to this as monoallelic expression because only one of the 2 alleles present in the offspring is actually going to be expressed. And dependent upon the gene, it determines whether it is the maternally inherited or paternally inherited allele that's actually expressed in that individual. So this genomic imprinting is permanent in the somatic cells of an animal. However, the marking of the alleles can be altered from generation to generation. So in that sense, this is a phenomenon of the germ cells in terms of the altering of that marking pattern. It can involve a single gene or an adjacent gene, ADJACT. It could be a part or the entire chromosome. And it could even include all of the chromosomes from one parent. Depending on their different species can have different patterns of genomic imprinting. So genomic imprinting can have a huge influence on human diseases. So here's some example of some mammalian genes that are actually known to be imprinted. And at this counting, or this year, the estimate is that approximately 1 to 2% of human genes are subjected to genomic imprinting. Here in this, previously, I talked about imprinting in mice involving the IGF or insulin-like growth factor 2 gene. That was a case where the paternal allele was actually expressed. Here I'm going to talk about a situation where you've got two genes that are under the influence of genomic imprinting, one in which the maternal allele is actually expressed, and a second one in which the paternal allele is expressed. Now, in terms of these 2 syndromes, Prader-Willi syndrome and Angelman syndrome, these are characterized, well, let's start with PWS first. PWS occurs in approximately 1 in 25,000 births or individuals, whereas Angelman syndrome or AS occurs in 1 in 15,000. So these are not very common, but they're not real rare. In individuals that have Prader-Willi syndrome, they have reduced motor function, obesity, and typically tend to have small hands and feet. In individuals that have AS, their characteristic symptoms are hyperactivity and thinness, unusual seizures, repetitive symmetrical muscle movements, and then also some degree of mental deficiencies. Now, typically PWS and AS involves a small deletion in chromosome 15. So if you inherit the chromosome 15 that has this deletion from the mother, you typically develop AS. If you inherit this chromosome 15 with a small deletion from the father, you develop PWS. So this deleted region is really critical to know where it comes from, whether it comes from the mother or whether it comes from the father. Having this deleted region is a big problem, period. But in combination with this phenomenon of genomic imprinting, it will lead to AS or PWS typically. So for the pattern for AS is because of the lack of expression of a single gene, and that gene is UBE3A. So UBE3A encodes a protein that regulates protein degradation. So it's a marker. It basically tags proteins, and it goes through this ubiquitin labeling process, and where then you can just chew up these proteins and reuse some of the components. If in individuals that have, well, no, let me say it this way. UBE3, for UBE3, the paternal copy is actually what's silenced. So that's the one that's tagged with methyl groups. Therefore, the mother's UBE3 gene is what's typically expressed in the offspring. So for an individual to develop AS, they have to have two things going on. Obviously, first is this happens, the paternal methylation of UBE3A. However, what has to happen also is that there is a deletion in chromosome 15 of the inherited from the mother, okay? And that deletion has to be in the region where UBE3 is, where that gene is. So the result is you only inherit one copy of UBE3. If you get it from your father, it's effectively not there because it's methylated and it will not be expressed. And you get this deletion in the chromosome 15 from the mother, or you get chromosome 15 that has a deletion from the mother, then you don't have a functioning or a working UBE3 gene that can be transcribed to produce that protein. Now, PWS involves or results from the lack of expression of this gene, SNRNP. SNRNP encodes a small nuclear ribonucleoprotein polypeptide N. Now you know why we use abbreviations. This protein is involved in gene splicing. So it actually controls gene splicing. If you can't remove the introns from a pre-messenger RNA, then a correct polypeptide is not going to be produced from the mature messenger RNA. And actually, that pre-messenger RNA or that messenger RNA will never even leave the damn nucleus. Okay, so in this case of PWS, let's just start with this again. SNRNP, in all cases, it's the maternal copy that is actually silenced. So to develop PWS, again, where you've got you've got a lack of expression of the single gene. So to develop this, you have to have the maternal methylation, not that we want to develop PWS, of that gene. Then you have to inherit from the father chromosome 15 that carries or that has a deletion, carries a deletion of that same region where the SNRNP gene, where the SNRNP gene is located. And actually, these genes are very close to each other, so a single deletion in that chromosome 15 can wipe out both of these genes. Okay, so let's look at this a little bit more. So why we're talking about this is because if you want to predict the phenotype of the offspring, you have to know first the imprinting pattern for these genes, i.e. in which parent that gene is actually going to be silenced, because that's how it's going to be in the gametes from that individual. And then you also have to know which parent contributes the chromosome 15 that has a deletion of this region. So notice this in this diagram, we've got our deleted region here that includes both the AS and PWS genes. So on this chromosome, that's the only one that actually can produce either of these products, or actually has the two functioning genes. Now, let's look on this side and what leads to the development of Angelman syndrome. Here we have an egg that carries chromosome 15 that has the deleted region. That gets fertilized by a sperm. In that sperm, the genomic imprinting pattern is that you're actually imprinting or marking the AS gene, which is UBE3A. So that one is silenced in the sperm. If these gametes fertilize each other, now we've got only one copy of the UBE3, and it's actually turned off. So we don't have another copy over here in this chromosome because that region was deleted in the female. These individuals would then develop Angelman syndrome because they don't have a functioning version of UBE3 anywhere in their cells. Now over here, let's pretend like the egg gets the non-deleted chromosome, or contains the non-deleted chromosome. However, in the maternal pattern is to actually add methyl groups onto this PWS gene, which is the SNRNP gene. Therefore, SNRNP is silenced in the egg. In this sperm, we've got a chromosome 15 that has that region deleted. So now, in this fertilized egg, we only have one copy of both of these genes. However, the only one that's silenced is actually the PWS gene. So that means no expression of SNRNP, therefore you're going to develop Prader-Willi syndrome. So Prader-Willi, I don't like that this does AS and PWS, but if it helps you remember it, that works for me. So there's two things that got to happen. The genomic imprinting is going to happen, period. But it depends on who you get that deleted chromosome that has a deleted region from, whether you get it from the mother or whether you get it from the father. If you get it from the mother, you typically develop Angelman syndrome. If you get it from the father, you'll typically develop Prader-Willi syndrome.